Hello everyone, welcome to my channel BioFreeLearner. Today we will learn Plasmid, the cloning vector, why we use Plasmid in RDT and features of Plasmid. Plasmid is a extrachromosomal DNA within bacterial chromosome which generally replicate independently. It is small circular double-stranded DNA molecule. Mostly they are found in bacteria and some archaebacteria also have it. In this video, we will look at how the plasmids are useful tool for genetic engineering and what are the important features that make the plasmid as important cloning vector. In copy machine, we select the copy number and get it. In genetic engineering, we want our gene of interest is more number of copies. And how we get it? More number of DNA. So we put a vector generally in the plasmid, put inside the molecular copy machine, that means bacteria, and ask the bacteria to copy. Now the bacteria grow in the optimal condition temperature. The plasmid which hold the gene of interest also grows and also we have more number of copies of our gene of interest. And some of bacteria inherits low number of plasmid and some bacteria have high number of plasmid. So the copy number of plasmid is regulated, and if you know about more how the bacteria is regulated the copy number, it will be explained later. The plasmid copy number is important because the plasmid copy number is too high, it causes energy burden to the bacteria, and if it is too low, it also chance to lose the plasmid or gene of interest. Now quickly look at the key features of cloning vector. Ideally, cloning vector has low molecular weight, having easy to handling, resistant to shearing, and isolate and transformation into bacteria and also easy. It has selectable and distinguished marker, so it can be different from non-specific one. This is important site for origin of replication, where replication of plasmid starts and is regulated by several sense and antisense RNA. Apart from origin of replication, the important site is multiple cloning site. It has put your gene of interest. It has several different restriction enzyme. Some gives rise to sticky end and some gives rise to blunt end. The cloning workflow first step is the restriction digestion of the vector with specific restriction enzyme that present in multiple cloning sites. Similarly, you cut the gene with the same restriction endonuclease, then ligation steps come where you join the gene of interest and in the plasmid vector backbone and inserting the gene of interest in the plasmid backbone. Then transformation. You transfer the plasmid into the bacteria and ask the bacteria to grow in the interest in number. Now the bacteria grow in culture media and gene number increase. If generally this is not happen, what actually happens? Some of the bacteria taken of the plasmid when transforming it and which bacteria don't take it. So we need to distinguish the marker or the selectable marker. Simply, if you put the antibiotic in the bacterial culture media, it should die. Bacteria should die and not grow. Only the bacteria which have antibiotic resistant gene against the antibiotics we have added will grow in that media. And the plasmid, if you have antibiotic resistant gene, then it survive, and the bacteria does not have the antibiotic resistant gene, it will die. So we not take the bacteria which don't have the antibiotic resistance gene. But here the problem is not solved. The problem is that some of the plasmids self-ligated and not having our gene of interest. Then the thing is that how we differentiate those two, whether it has our gene of interest or self-ligated one, not taking our gene of interest. For that, we need screening markers. Mostly in the multiple cloning site, there are several genes. LAGZ is among one of them, which hydrolyze the lactose into glucose and galactose, allowing certain enzyme to the bacteria and use the lactose as an energy source. LAGZ is important component of lactoferrin, which contain the enzyme beta-galactosidase, and beta-galactosidase can convert the X-gal, the color product. If the bacteria transform into plasmid having LAGZ as a selectable marker, then the bacteria colony would be blue in color because beta-galactosidase is active and it converts the color into blue color. And somehow you insert the gene of interest in the manner and the LAGZ gene is disrupted and beta-galactosidase is functionless and you don't have the blue colony. You will get white colony. So the blue colony is self-ligated plasmid and the white colony contain our gene of interest. Now we will grow it in the culture media. Thank you for watching.